Welcome to the Absolute Antibody Prometheus 2.0 Humanization presentation. I'm Damian Leung. I'm the head of antibody engineering at Absolute Antibody. Prior to joining Absolute, I worked in pharma for 16 years and delivered several molecules into the clinical pipeline. I hope you'll find this webinar useful, and please feel free to reach out if we can help you out. So what is humanization and why is it important? In order to reduce the risk of immunogenicity when developing therapeutics for human, we need to maximize the humanness of the protein. If the initial binding antibody came from a non-human source, for example from mouse or rabbit immunizations, we need to humanize it by replacing the parental frameworks with human frameworks. It is, however, a fine balance and an art form to change the sequence while retaining the binding and the functional activity of the parental antibody. There are many FDA-approved treatments that are humanized antibodies out there. So how does our Prometheus humanization method stand out from CDR grafting? At Absolute Antibody, we focused on the manufacturability of the antibody. We selected a set of diverse human antibody frameworks that are known to have good expression profiles and have been used in clinical molecules. We use in silico modeling to identify key structural residues that are crucial for the presentation of the CDR loops. So that we can have the correct paratope for binding. We then produce a matrix of heavy chain and light chain combinations to find the best clones that are high yielding with comparable affinities to the parental antibody. This is a case study with 25 variants that we produced in a humanization campaign. The relative expression levels are shown on the left axis and as bars. From the left to right, the black bar is the chimeric antibody. Blue, yellow, green, and red are different humanized variants. And as you can see, most of the clones have higher expression than the chimeric antibody. Some of them are more than 20% higher. The purple dots represent the monomer content in percentages on the right axis. The chimeric antibody in the far left has about 7% aggregate. And here you can see the diversity in profile between the clones. Some of the good expressors actually have more aggregates, such as the one in green. Which is interesting because they have similar heavy chain, while the samples in yellow have similar light chains. And the red samples has a combination of these unfavorable heavy and light chains. It's not surprising, though, that the heavy chain has a bigger influence on expression than the light chain. We're here to share that we've successfully humanized rabbit and camelid antibodies with the launch of Prometheus 2.0. And as we all know, rodent antibodies are more straightforward to humanize because of their structural similarity to human antibodies, while rabbit and camelids can be more challenging. Rabbit antibodies have longer CDRs and sometimes are disulfide bonds between the variable and constant region, while the camelid antibody can exist as single domain binders that don't even need a light chain. In this example, rabbit alpha-5 beta-5 integrin antibody was humanized, and we produced 4 IgG variants. And we compare the target binding by ELISA. And as you can see, 3 out of 4 of the variants have almost identical affinities as the parental chimeric antibody. In this example, we took a camelid anti-HER2 VHH and produced the human chimeric IgG fusion as a control. We then designed 2 humanized VHH variants and compared the binding of these Fc fusion proteins to the chimeric. And interestingly, you can see one of the variants appeared to have even better binding than the chimeric. So to wrap up, at Absolute Antibody, we've launched Prometheus 2.0. Although non-murine antibodies such as rabbit and camelids can be more challenging, we were able to successfully humanize them and retain binding. Thank you for watching this webinar. We look forward to help our client engineer the best molecule possible. Bye.